Good morning to all. Today we had to discuss about polymerase chain reaction. The polymerase chain reaction is nothing but it is a technique. It is a simple laboratory method for in vitro DNA synthesis. This technique is used for amplification of specific DNA fragments when their number is low, very low in a complex mixture. The method was The method was invented by Gary Mullis in 1980s. Before PCR, DNA of interest could only be amplified by overexpression in cells with this limited yield. Polymerase chain reaction amplifies known sequence of DNA to several orders of magnitude. Requirements magnitude. A specific DNA fragment to be amplified, that is DNA template. A template DNA can be genomic, plasmid or cDNA. The polymerase enzymes will read and copy this DNA. One DNA molecule is used to produce multiple copies exponentially. DNA polymerases will add nucleotide to the 3 of this primer according to the normal base pairing rules. PCR typically amplifies fragments between 0.1 to 10 kilobase pairs. PCR consists of a series of 20 to 40 cycles. One PCR cycle comprises of 3 steps. First one is denaturation, annealing and extension. In these steps, initializations, temperature used and the length of time applied in each cycle depend. TM primers, DNA polymerase, DNTPs and divalent ion concentration. Initialization steps. In these steps, the reaction is heated 94 to 96 degrees Celsius for 30 seconds to several minutes. This step is usually only done in the very beginning of the PCR reaction. The step is important for activating hot start polymers if we use such polymers and to denature your template DNA. If the template GC content is high, we may need to perform an extra long initialization step. Heating dissociated the inhibitor DNA polymerase complex. Step 1, denaturation. First cycling event, heating reaction mixture 94 to 96 degrees Celsius for 30 seconds. DNA melting by disrupting the hydrogen bonds between bases yields single stranded DNA. Step 2, annealing. Reaction temperature is lowered to 50 to 65 degrees Celsius for 20 to 40 seconds. The temperature in this step needs to be low enough that the denatured primers can form Western Crick base pairs with the template DNA, but high enough that only the most stable, perfectly paired double-stranded DNA structures can form. Usually, this perfect annealing temperature is a few degrees lower than melting temperature of the primer pair. During these steps, the polymerase will bind to the primer template's DNA complex, although the polymerase will not start recording until the temperature is raised in the next step. Step 3 is extension. The temperature depends on polymerase used. Tag polymerase has its optimum activity temperature at 75 to 80 degrees Celsius. DNA polymerase synthesize new DNA strand complementary to the DNA template strand by adding DNTPs. The extension time depends on DNA polymerase used as well as length of DNA fragment to amplify. At its optimum temperature, the DNA polymerizes 1,000 bases per minute. PCR steps: denature, anneal, extend pairs and cycling repeat. Denature double-stranded DNA, anneal primers to a single-stranded DNA extend primers yielding a new double-stranded DNA. The cycling repeat steps 1 through 3, 20 to 30 times. PCR requirements: DNA template containing desired segments, primers, DNA polymerases, deoxynucleotide triphosphates, buffers, bivalent cations, magnesium and manganese, monovalent cation, potassium, and additives. Then thermocycler. Earliest thermocyclers were designed to with Glenov, fragment of DNA polymerases, platter element. Modern PCR machines did temperature on to 105 degree Celsius. Components of PCR. Procedure. The procedure for typical PCR is mentioned as below: denaturation of double-stranded target molecule, renaturation annealing of primers with complementary region in DNA template strand, synthesis of a new strand. Components of PCR. The PCR region requires the following components: DNA template, the double-stranded DNA of interest separated from the sample, DNA polymerase, usually the thermostable tag polymerase that does not rapidly denature at high temperatures 98 degrees Celsius and can function at a temperature optimum of about 70 degrees Celsius. Oligonucleotide primers, short pieces of single-stranded DNA, often 20 to 30 base pairs, which are complementary to the ends of the sense and antisense strands of the target sequence. Deoxynucleotide triphosphates, single units of the bases A, T, G and C, DATP, DTTP, DGTP or DCTP provides energy for polymerization and the building blocks for DNA synthesis. Buffer system includes magnesium and potassium to provide the optimal condition for DNA denaturation and renaturation. Also important for polymerase activity, stability and fidelity. Types of PCR: reverse transcriptase PCR, real-time PCR, assembly PCR, asymmetric PCR, colony PCR, etc. First one is reverse transcriptase PCR. Detect gene expression through the synthesis of complementary DNA, cDNA transcript from RNA. RNA template is converted into a complementary DNA using reverse transcriptase, primers, oligo random primers and gene specific primers. 1 step and 2 steps RT-PCR. Amplified DNA fragments that are produced can be analyzed by agrosgel electrophoresis. Amount of amplified fragments produced proportional to the amount of targeted mRNA in the original RNA sample. Real-time PCR or qPCR. The RT-PCR process amplification of DNA is monitored in real time. PCR with an added probe or dye to generate a fluorescent signal from the product. Detection of signal in real time allows quantification of starting material performed in specialized thermal cycles with fluorescent detection systems. PCR signals is observed in an exponential curve with a lag phase, log phase a linear phase and a stationary phase. Third one is assembly PCR. Formation of large oligonucleotides DNA from short segments. Each oligonucleotide is designed to be either part of the top or bottom strand of the target sequences. An oligonucleotide anneal to complementary fragments that and then are filled by polymerase. Each cycle thus increases the length of various fragments randomly depending on which oligonucleotide which find each other. Production of synthetic genes and even entire synthetic genomes. Fourth one is asymmetric PCR. Amplifies one strand of the target DNA used in sequencing and hybridization probing amplification of only one of the two complementary strands is required. PCR is carried out with a great excess of primer for strand targeted for amplification. Colony PCR. Screening of bacteria, Escherichia coli or yeast clones or correct ligation or plasmid products. Individual transformants can either be lysed in water with a short heating step or added to the PCR reaction and lysed during the initial heating step. Initial heating steps causes the release of the plasmid DNA from the cell so it can serve templates for amplification reaction. Primers designed to specific target the insert DNA can be used to determine if the construct contains the DNA fragments of interest and also insert orientation. Hot start PCR, inverse PCR, nested PCR and touchdown PCR. Applications of PCR, molecular identification, sequencing and genetic engineering. Molecular identification, molecular archaeology, molecular epidemiology, molecular ecology, DNA fingerprinting, classification of organisms, genotyping, prenatal diagnosis, mutation screening, drug discovery, genetic mapping, direction of pathogens, sequencing, bioinformatics, genomic cloning, then genetic engineering, site directed mutagenesis applications and site mutagenesis gene expression studies. Cystic fibrosis can use mutations in the cystic fibrosis transmembrane regulator CTR of genes. If non-CF individual CTFR gene codes one protein that which is that with applications the PCR widely used in cloning for short DNA fragments obtained genomic or cDNA clones. The chromosomal DNA is first isolated and purified from the cells. Then the strands are separated by brief heat treatment. This is followed by the addition of oligonucleotide primers that hybridizes with the complementary sequences and of DNA strands. Several cycles of PCR reactions takes place leading to amplification of DNA fragment between the two primers. PCR used in the detection of infectious pathogenic microorganisms. The various steps involved are the follows. The blood sample from the infected person is collected in a test tube. The tubes are centrifuged and settled down while the serum floats above. The viral RNA genome of HIV particles is extracted from the serum of the infected person. An oligonucleotide primers is then added. This is followed by incubation of reaction mixture with enzyme reverse transcriptors and 4 types of deoxyribonucleoside triphosphate is followed by PCR amplification. Finally, the viral genome is run in gel electrophoresis and compared with that of control using blood from non-infected person. PCR is used in the generation of DNA fingerprints in the forensic medicine in order to scientifically investigate the case of injury and death in unexplained circumstances. It is used for the identification of criminal and legal cases, disputed parentage. PCR is also used in gene therapy. Thank you all.